Respected faculty members, dear students and viewers from all parts of the country, welcome to the public lecture series organized by the Department of Microbiology, Surendranath College. On behalf of organizing committee, I welcome you all to the 3-week-long webinar series titled COVID-19 Second Wave, Evolution, Risk and Vaccines. The webinar will also be broadcast in the official YouTube channel of our college on every Wednesday in the month of May 2021. I am your host, Devdeep Dasgupta, a faculty member at the Department of Microbiology. As per the recent data and statistics published in the journal The Lancet, the scenes of suffering in India are hard to comprehend. As of now, May 4th, more than 20.2 million cases of COVID-19 has been reported, with a rolling average of 378,000 new cases per day, together with more than 222,000 deaths officially. In this frightful background, Department of Microbiology, Surendranath College has come forward with a webinar series to understand the evolution of SARS-CoV-2 virus, its double mutant variant, the impact of second wave, and finally about the vaccination process for building up the immunity against COVID. A sincere welcome to all those of you who are watching us online through either Zoom, YouTube, and Twitter. Before going into the detail, I would like to request our Principal Sir, Indralil Kaur, to deliver the inaugural note.
Thank you, Devjit. Am I audible?
Yes,
Hello.
sir. Yes, sir.
Yes, sir. Yes, sir.
Thank you. So I, on behalf of Surendranath College, am elated to thank all of you who have joined this platform to understand the happenings around us in relation to COVID-19. I also thank my Department of Microbiology and Molecular Biology who have taken the efforts to organize this webinar to enlighten all those concerned about the present happenings in and around our country and the globe. COVID-19, second wave, evolution, risk, and vaccinations are very pertinent thoughts around in this COVID-related situation. Today we have Professor Dhrubajyoti Chattopadhyay, Vice Chancellor of Sister Nivedita University, among us to enlighten us about the various aspects of this COVID-19 related second wave, evolution, risk, and vaccination. Before I go into that, I request, I just speak a few words about this present pandemic situation. If we have learned from Dr. Somas Swaminathan, who is the principal scientist of WHO, she has really warned us regarding the second wave and the future waves to come. And she has also pointed out in our preparedness, the lacunae in our preparedness particularly. And that is how every day we are really scared to look at the newspapers which come around for us. And we see large number of dates, funeral, pyres, and all these are burning together and making us really, the people around the entire country are really scared at this moment. We do not know what will happen to our second or the third wave or the future waves to come around. We know the virus has really become very smart in changing itself to cope up with the various changes and to give its own fighting with the preparedness of the humankind. Hereby, we are really in a very controversial state where we do not know the reach of vaccination, the vaccination and its impact, the risks of various inoculative subjects wherever we are now going. Some entire country is suffering with partial lockdown, full lockdown, and various other methods. Some stopgap methods which are being employed to slow down the process of inoculation and so that our preparedness can also help in the fight with the COVID-19. I understand that the entire platform will be really looking after they will be really enlightened to understand that Professor Dhrubajyoti Chattopadhyay will tell them regarding the present situation and the condition thereafter to fight out the COVID-19 epidemic. I should say pandemic. And this is a series of webinars which are going to enlighten us through these next 3 weeks. Today is the first lecture, and we'll be having the lecture every day, every Wednesday from hereafter in this platform, and hope this will, at the end of this lecture, will be really enriched and have garnered enough confidence to fight out with this COVID-19 pandemic. With these few words, I welcome Professor Dhrubajyoti Chattopadhyay to this platform, and I hand over the mic to the HOD for further discussion.
Thank you, sir, for your inspiring note. Today we have with us Professor Dhrubajyoti Chattopadhyay, Honorable Vice Chancellor of Sister Nivedita University and ex-Pro Vice Chancellor of Academic Affairs at University of Calcutta. He was also the Vice Chancellor of Amity University, Kolkata campus. It is my distinct pleasure and honor to introduce him today. Professor Chattopadhyay had been the catalytic force for nurturing modern biological science research and teaching in Eastern India since early 90s. He graduated from Department of Chemistry and Presidency College, followed by post-graduation from Department of Biochemistry, University of Calcutta. He earned his PhD from Bose Institute in the year of 1984, followed by his postdoctoral research work in Roche Institute of Molecular Biology, where he studied vesicular stomatitis virus. Thereafter, he studied negative sense RNA viruses at the Cleveland Clinic Foundation, one of the pioneer research institutes in the United States. He continued his research work in several universities thereafter in the USA and Japan. Professor Chattopadhyay joined University of Calcutta and elected as a Guha professor at the Department of Biochemistry in 1997. His research work includes works on Vibrio cholerae, Giardia lamblia, negative sense RNA viruses, and also cell signaling and enzymology. He was elected as the Fellow of National Academy of Science in 1998, Indian Academy of Science in 2004, Fellow of West Bengal Academy of Science and Technology in 2006. Dr. Chattopadhyay had lifetime membership in various organizations and scientific associations, including Indian Science Congress Association, Asiatic Society, Biotechnology Society, Virological Society, to name a few. Above all this, Professor Chattopadhyay is a wonderful teacher. He has a glorious teaching experience for more than 35 years, during which many students have completed their PhDs successfully under his able guidance, and has brought fame and glory to themselves as well as to our country. His work is recognized in more than 150 research papers in the top research journals, including Cell, PNAS, Journal of Biological Chemistry, Journal of Molecular Biology, RNA Biology, and several book chapters to his credit till date, with more than 3,500 citations. Professor Dhruvajyoti Chattopadhyay is a scientist and academician par excellence. Today, he is going to discuss the key aspects of this deadly catastrophe of COVID-19, including its origin, evolution, immune response, life cycle, and finally, the emergence of SARS-CoV-2 in India and important information on vaccination strategies. Saying that, I now request Professor Chattopadhyay to take the mic.
Thank you, Professor Devdip Dasgupta, for your very elaborate introduction. I really want to congratulate Dr. Indranil Kaur, the principal of Surendranath College, who actually spearheading the college to get the academic excellence and looking after the quality of education also. Dr. Kaur, I'm also very happy that I was actually associated with the establishment of the Department of Molecular Biology and Department of Microbiology in Surendranath College. And now I could see that from those days, it matured into one of the best departments in this particular area in this part of the country. Now, that's only possible because of you have a very distinguished faculty members, and they are trying their best to give the students the best exposure. At this particular juncture, to organize this seminar, a month-long seminar, and in each week you are identifying the different people who are working with in this field is something which is very remarkable. Thank you very much, Dr. Kaur, Dr. Dasgupta. And also, I am very happy to have my students who are faculty members, and definitely all the student participants who have joined this particular seminar. Now, when Devdip actually contacted me regarding this seminar, the title itself is something which gives a huge background for this present crisis. The reason why I'm going to tell you, you know that the reason the virus always excited me is that it is that material which is in the borderline of living and non-living. So virus itself is a non-living material, so only becomes living when it comes into contact with the living cells. So it needs a host, otherwise it's just a complex chemical. So from the very beginning of my career, research career, I took virology as one of my research area, and my PhD in Bush Institute is actually in relation to this. One of the, I should say that virus, which is a bacterial virus, which we call as lambda virus. Later on, I moved to postdoctoral to get the study on the RNA viruses, which also intrigues me. And when this pandemic started, I could find it out. It is that RNA virus which is making so much of havoc. And if you look at the history, the last few pandemics, all are due to the different RNA viruses. Smallpox is a DNA virus, but after that we have the flu pandemic, we have this one after another pandemics of flu, the last one in 1980, the Spanish flu. And in all these cases, the viruses and this RNA viruses actually played a very important role. And in the last 3 decades, HIV played a very important role. So coming to this COVID-19, if I could have my first slide. For my dear students, I just want to mention that if we look at the viruses from the broader, yes, next slide, please. Next slide is fine, yeah. So the order of the virus is very important. So it is actually belongs to the nidovirales, where in one of the family member in the nidovirales is Coronaviridae. And in the Coronaviridae family, also there are two subfamilies, Toroviridae and Coronaviridae. And this subfamily Coronaviridae is again subclassified into 4 different genus. And these genus are alpha coronavirus, beta coronavirus, gamma coronavirus, delta coronavirus. Now, I have given the examples of the different alpha, beta, gamma, and delta. But if you look at the beta coronavirus, the lineage, so first comes order, then comes family, then subfamily, then comes a genus. And from genus, we could also classify them into the lineage. The beta coronavirus is actually 4 lineages are there, lineage A, B, C, and D. And lineage A has got the human coronaviruses. Lineage B has got the SARS-CoV and the MERS-CoV. Lineage C has got the BT-COVID, and lineage D also have got another BT-COVID. The avian coronavirus belongs to gamma, and in this way, our interest is the beta coronavirus lineage B, which is the SARS-CoV. The lineage C has got the MERS, that is, which are actually the camels are there. Next slide. So the first wave, which initiated a massive global effort to understand the science behind this deadly virus, which officially we said that we started in the December 2019 and the first death in January 2020. But now we could actually, after going through more information, there is a possibility it started earlier than that. Next slide. When the structure of this SARS-CoV-2 and its key antigenic components, if we look at it in this particular slide, now the SARS-CoV-2 is a single-stranded RNA virus, and the key antigenic components being targeted for the vaccine development is shown to the right, consisting of the spike protein, the envelope protein, the membrane protein, and the nucleocapsid N proteins. Now, the envelope, the main emphasis for the human vaccines is based on the envelope protein and consisting of an S1 binding region and S2 fusion and entry region. And that itself is actually giving us this, the S1 domains important. The S1 domain, it contains a receptor binding domain and is responsible for binding to the AC2 receptor on the surface of the cells. Now, following fusion, the S protein sheds the S1 region, and there is a dramatic structural change to its post-fusional states in order to the virus to enter into the host cell. So this is how actually we could find out that these antigenic components and their role in entering into the host cell. Next slide. Now, the proposed SARS-CoV-2 transmission routes, what we are right now looking at, is that if the SARS-CoV-2 and the infected patient, if they're coughing, the aerosol, which is actually of less than 5 micrometer diameter, this actually goes to the ear and it can move up to more than 1 meter. And that's why some confusions are there. These aerosols less than 5 micrometer diameter are actually giving rise to going to the uninfected patients and they get the infection. Besides that, the respiratory droplets are there, which are greater than 5 micro diameter. They form the droplets. They can not travel more than 1 meter, less than 1 meter, and then it can adhere to the different surfaces. It can actually associate it with our hands and gets in direct contact. It actually added to the surfaces, door handle, glass slab, cell phone, countertop, and all this gives rise to indirect contact. So this particular part is very, very important. So for airborne infection, which are actually the aerosol less than 5 micron diameters, which can move more than 1 meter. Now we are talking about, as Principal Science mentioned also, the Swaminathan's recent announcement that this 6 feet, more than 6 feet distancing is very, very important also. And also sometimes it happens that it can also go through the our fecal route, where the droplet nuclei are there, and the fecal oral is another particular infection domain. So in that way, the transmissions can be of various types, and you can get the different transmissions. But the dashed line shows the plausibility of transmission types that are actually what we are talking about airborne. So when we said airborne means it's actually coming from the infected person, but it forms a smaller aerosol which can travel longer distances. Next slide. Now, when we talk about these clinical symptoms of this COVID-19, now COVID-19, it actually manifests in different ways. There are respiratory tract manifestations as well as the extra respiratory manifestations. When you talk about the respiratory tract manifestations, normally it goes to the nose, rhinacea, anosmia, pharynglia, and rhinoglia. And then it also has sinusitis. It can go into the mouth, ageusia, and the throat, which causes sore throat, the pharyngitis. And then it can go to the lung, the cough, dispenses, expectorant, chest pain, pneumonitis, ARDS. So in that way, we can have the respiratory tract manifestations. And the extra respiratory manifestations are the neurological, the dizziness, the headache, the implanted consciousness. We have also the muscular, the myalgia, arthralgia, malaise, neck pain, back pain, the lung, the heart, where with that arthmesis, the chest pain, and the digestive, which causes diarrhea, nausea, vomiting, abdominal pain. But there are systemic symptoms also, like the chills, the fatigue, the malaise, the fever. So these are the different types of manifestations: respiratory tract, extra respiratory tract, and systemic symptoms. Next slide. So these are the clinical symptoms. Now, if we look at the lung pathology under this, the following, the inhalation of the severe acute respiratory syndrome coronavirus 2 into the respiratory tract, the virus traverses. That is actually what we could see. The virus traverses a huge amount of distances, and we can get that whole area. Can I have the screen, please? Yeah. The virus traverses just in the lower lung, where it infects the cells, including the alveolar and the epithelial cells, the vascular endothelial cells, and alveolar macrophages. Now, upon entry, the SARS-CoV-2 is likely detected by cytosolic innate immune sensors. This is very important. Please understand this. It's detected by cytosolic innate immune sensors as well as endosomal toll-like receptors that signal the downstream to produce the type 8 interferons and pro-inflammatory mediators. Now, the high concentration of inflammatory cytokines, the chemokines, amplifies the destructive tissue damage by endothelial dysfunction and vasodilation, allowing the recruitment of immune cells. What type of immune cells? Macrophages, neutrophils. Now, the vascular leakage and the compromised barrier function, it promotes the endothelitis and the lung edema, limiting gas exchange that then facilitates a hypoxic environment, leading to respiratory organ failure. The inflammatory milieu induces endothelial cells to upregulate the leukocyte adhesion molecules, thereby promoting the accumulation of immune cells that may also contribute to the rapid progression of respiratory failure. Now, the hyperinflammation in the lung further induces the transcriptional changes, and these changes are actually what we could see at the end of this function. So we can divide this particular COVID-19 infection in 3 different stages: the phase 1, phase 2, and phase 3. So the right after infection in the phase 1, it could be arrested. If it goes into the phase 2, then this particular cytokine, then it actually gives rise to this cytokine, and the cytokines, if it can also be arrested over there, but if it goes to the phase 3, you can see that immediately you can get the cytokine storm, and that's the most critical stage. Next. So in the life cycle, more or less what we could see, the virus entered, as I mentioned to you, ACE2 receptor, and there are one protease, and these two actually changes the structural proteins, and then it actually enters into the cell, and then it goes into the lysosomal, the cathepsin L, and then it gets released into the system. We get this particular RNA which comes out, binds the ribosome, and you can get the different proteins are being translated, and you can see the proteins are actually there, which synthesizes the polymerase and other proteins, which helps the RNA to replicate, and we have the RNA replication going on. So both the genomic as well as the subgenomic RNA are being produced, and you can get this particular subgenomic transcription followed by translation, and ultimately we could have the assembly and budding out of this, which we call as a release of this particular viruses. So this whole mechanism coming inside to the release is actually what we call as a SARS-CoV-2 life cycle. And you can see there are various points. That means at the entry point, you can actually stop it. If we actually have something which is not going to allow them to bind to the ACE2 receptor or the protease to function to leave that and change the structure, it can actually act on the viral polymerase, which can inhibit the replication of the virus. And then you can see that it can also try to affect this transcription translation process, or ultimately it can the processing process, which also inhibit the release. So in all different places, the people are trying to develop some things which can actually act as this antivirals or work against the virus. Next. So once we know about this, so in one, the cell goes inside the cell. So what I first tried to actually let you know about, that what is a lung pathology, now the virus cell life cycle, and thereby the two are coming together. And I mentioned to you the innate immune response is actually been demonstrated by the dendritic cells and the monocyte macrophages. The antigen presenting cells such as monocytes, macrophages, dendritic cells, which recognize the pattern associated molecular patterns. Remember, this is a very important term, PAMPs, pattern associated molecular patterns, which expressed by the SARS-CoV-2 by the pattern recognition receptors, PRRs, and just like the TLR3 and 7. Now, these activate the intracellular signaling pathways leading to the expression of type 1 and 3 interferons, which in turn activate the innate immune cells to produce pro-inflammatory cytokines and chemokines. This leads to an influx and activations of neutrophils, and further, the antigen-presenting cells and other innate immune cells, such as the natural killer cells. So this is what we call as the innate immune response. So phase 1, if with this innate immune response, we can actually control the virus, then the virus cannot move on to the phase 2 stage. Next slide. Then the adaptive response comes. The adaptive response is activated following the viral uptake and the antigen presented by a range of APCs. The antigen presenting cells, the antigen to B cells, which then differentiates into antibody producing plasma cells. The neutralizing antibodies then bind to key viral proteins, such as the spike protein, and neutralize their activity. Now, when we talk about the rapid antigen stage, the test, we are talking about identifying this neutralizing antibodies, which comes in this phase. Now, other antibody-mediated antiviral functions include the antibody-dependent cellular cytotoxicity. This is ADCC, antibody-dependent cellular phagocytosis, ADCP, and antibody-dependent complement activation, ADCA. So there are three different types of categories: CC, CP, and CA. Now, the cytotoxic CD8 plus T cells, they kill virally infected cells via the production of enzymes and perforin and the expression of the Fas ligand, all of which mediates cellular apoptosis. A series of CD4 plus T cell populations are involved in the adaptive cellular response to SARS-CoV-2. The follicular helper T cells and the Th2 CD4 plus T cells both provide help for B cell antibody production. Th1 and Th17 CD4 plus T cells are also thought to play a key role in the inflammatory response and viral killing.
Hello.
What happened?
T cells have been implicated with an immunoregulatory role in SARS-CoV-2 infection via the production of anti-inflammatory cytokines and contact-mediated cellular suppression. Whether CD8 plus Tregs and Bregs play a role is not currently known. So this is how actually we look at the adaptive immune response. I hope I could explain to you the two phases. One is the innate immune response, and the second one, which we call as an adaptive immune response. Now, innate immune response is very important to control as a phase 1 of infection, a little bit to the phase 2, adaptive immune response in the phase 2. But if this is not being there, then it goes into the phase 3, where the cytokine storm starts, and there will be very difficult actually. Next slide. So the cytokine storm and the T cell lymphopenia is associated with this COVID-19 severity. Now, this is actually, this slide shows you that actually what is actually when we are in the phase 1, there are no symptoms. And then in the phase 2, what happens? There are this cough, temperature, and other things. And the last one, actually, when we call the severe, the phase 3, is actually where we can get this whole range of the different types of cytokines, which are actually becoming very difficult to control. Next. This is one way when we look at the immunity. But another very, very important work has been done recently, and in 2021, there are some very important papers published this year in the month of January, February, March. Now, this is a SARS-CoV-2 human protein-protein interaction network. Why it is important? Because we wanted to know that in the gradient of the different population, the SARS-CoV-2, its function, its effect of its infections, it varying very much. So the network representation of the high confidence SARS-CoV-2 host interactome has been studies in the HEK293 cells and taking the SARS-CoV-2 beta proteins and interacting host proteins. And then it has been shown that what happens, the cytosol in the nucleus, in the mitochondria, in the cytoskeletons, the Golgi, the plasma membrane, endoplasmic membrane, endosome, as well as in the ultimately for this particular localization of this, which are still unknown. So this is a complicated slide, but what I want to say that this is a very important slide for identifying how to approach in future for fighting with SARS-CoV-2. Next. To the 4 heartful feedback loops in the pathophysiology of COVID-19 is actually going to be the one is that, there is increased ACE and the angiotensin, where that actually platelet dysfunction is going to be there. There will be no PGR2. The cytokines, chemokines goes up, and there are vascular leakage. Now, this will happen if you look at the evading the interferon response, the SARS-CoV-2 infection, where the ACE2 is downregulated. And what happens? This SARS-CoV-2 infection gives rise to free virus replication to the PAMs and NAMs, which again evades the interferon response. If you look at the AECs and ECs, the apoptosis and the pyroptosis, there also we could see that cytokines, chemokines upregulated. We find the T-cell lymphopenia, and the infiltration of the macrophages, PMNs, or the increase in the cytokine chemokines, how they are actually T-cell lymphopenia is going up. And ultimately, if you look at the EC's dysfunction, we have the different types of organ failures, the platelet dysfunction, the thrombin generation, and from the tissue factor also we can get the thrombin generation. So in all, we can see that these damage-associated molecular patterns, the DAMPs, are there very important, and it's getting important to look at this, the DAD, ALI, the ARDS, and EOT. You can find out these are the 4 heartful feedback loops in the pathophysiology of COVID-19. Next slide. In this background, I come to this vaccination. Why vaccination is important? Vaccination is very, very important. Next slide. Now, the COVID vaccine, if we look at it, the vaccine shots are one of the tools we have to fight the COVID-19 pandemic. And there are, even if we have these vaccine shots, the masks are very important. The distance is also very, very important. So we have to make the vaccination very effective one if we still carry on with the masks and the distance. So we have to understand the vaccines are safe because it will only come after a huge amount of the background work. And there are different types of COVID-19 vaccines are right now available, and many new vaccines are also going to come. Next slide. Now, the vaccine platforms which are being employed for the SARS-CoV-2 vaccine design are of different types. You know that we have this protein and the peptide subunit vaccines, which are actually cytotoxic. I mentioned to you, cytotoxic T cell, dendritic cell, using that immune cell therapy could be done. There are nucleic acid-based vaccines, mRNA vaccines or DNA vaccines. We know the mRNA vaccine is already there, and the DNA vaccine is actually coming up by one of the Indian company, Zydus. And then the attenuated virus, the whole virus vaccine, the attenuated or the inactivated viruses, the whole or the subunit vaccines, which also has the Covaxin, the Bharat Biotech, which came up with the NIV and ICM together. And that is actually there for a long, long period of time with many other viral disorders, for example, the polio. And the other one is the viral vector vaccines, and that also can be of 2 types. One we call as a non-replicating viral vector, another is a replicating viral vector, and any one of them can actually give rise to this as a vaccines. And the nanoparticle and the virus-like particles vaccines are right now we are talking about also. Now, this viral, the VLP vaccines can also have antigen attached to their surface or combined in their core. The immune cell therapy approach uses genetically modified SARS-CoV-2 specific cytotoxic T cells and dendritic cells expressing viral antigens to protect against the SARS-CoV-2 infection. Now, each of these vaccine approaches has benefits and disadvantages in terms of cost, ease of production, safety, and immunogenetics. And it remains to be seen which of the many candidates in development protect against this COVID-19. So at the very beginning, I find that Nilangsha has got one question: how long these vaccines are going to be effective? For 6 months or thing? See, vaccination started very recently, and actually the information regarding how long it will be effective, it's still we have to wait for a couple of more months, because it is very difficult from the experimental to when we go to the population, that particular vaccine, how long it's going to be there. Now, next. So the mRNA COVID-19 vaccine, how it works. You people, another question is I find that how the different platforms are working. So let me try to explain to you about this, how mRNA COVID-19 vaccines work. Now, understanding the virus that causes COVID-19 is, so you know that the mRNA is there, and in this vaccination, what we can do, that how we have to understand how does this vaccine in actually gives your cells how to make copies of the spike proteins. If you're exposed to the real virus later, your body will recognize it and know how to fight it out. So the mRNA belonging to the gene sequence of the spike protein. And if it goes inside the cell, it will synthesize this and it will get it. Now, the viral vector COVID-19 vaccine works in a completely different manner. So the coronavirus, like one that the COVID-19 named for the crown-like spikes on the surface, and the viral vector vaccine uses a harmless version of a different virus called a vector. So, for example, adeno platform may be used, which is actually we found in case of this AstraZeneca one, or the vesicular stomatitis can also be used. The vaccine teaches us that how actually we are going to get the things one after another, and this particular thing is going to help us to give rise to this, the replicating vector or the non-replicating vector. Next. The second wave. The second wave has hit us hard. Just when we were almost getting ready for the final battle, you know, the whole COVID infection going down, and this Lancet paper where the principles are already mentioned, that which showed that that particular complacency is one of the most important reasons for India to have this terrible second wave, and which actually also happened in case of US, UK, and European countries. Next slide. So this is one, you could see that this is already changed. This is up to April 22, but we know that it's already changed and it's going on more and more and more. Next slide. So this emergence of the SARS-CoV-2 variants is very important to understand. You know, the B1.1.7, this is first detected in September 2020 in UK. We call it the UK variant, and detected in other countries. And then we could see, we call that as a UK strain. It has got an increased transmissibility. Then we have the B1.351, first detected in October 2020 in South Africa. we call it a South Africa strain, and also detected in other countries. So UK variant is more than 50 countries, and South African variants is less than 20 countries. And there also increased transmissibility and possible reduction of vaccine effectiveness. Then we have the third one, the name P.1, first detected in December 2020 in Brazil. We call the Brazil variant. And it was detected in other countries, but not like the UK or the South African variant. And it also has got the increased transmissibility and possible reduction of vaccine effectiveness. Now, if now I can tell you the V1.1.7 UK variant, the Brazil variant, the South African variant, the latest one addition is the double mutant B1.617. This has got mutation which is similar to another variant, the E484K. The E484K found in the first spread in Brazil and South African regions. Now, it includes the L452R mutation, the second mutation, which helps the virus escape the body's natural immune response. So you understand that natural immune response can restrict it to the 1 or the 2, so that the cytokine storm is not going to be there. But if it cannot restrict at those stages by lowering the body's natural immune response, then what will happen? The patients will show cytokine storm, and at that stage, so it will go fast to the phase 3, and at that stage, it's very difficult to control. And this variant has been detected in at least 10 other countries, including US, UK, Australia, New Zealand, all these places. Now, this is one which was first detected in Maharashtra also, and there is a huge amount. So we normally said this is actually the one which is mainly the UK variant we found mainly in Punjab area. The Brazil variant also in Delhi and Punjab. The South African variant we have seen it's in the Andhra Pradesh and the southern part of the country. And the double mutant is from Maharashtra to Bengal. And we could see that the things are becoming more and more complicated. Next one. So implications of this emerging SARS-CoV-2 variants, it's that antibodies induced by vaccination or previous exposure to a virus, it actually gives a chance that normal viruses are there and there are virus variants, because though we found that there are some checking activity in the COVID-19, replication checking activity, but it's not so strong. Still, there are mutations, and those mutants are actually not fully neutralized and may not be fully neutralized in case of the variants. So what happens? That this 1-2 phase is being crossed by these variant viruses, and that's why it's getting very difficult. Next one. Next one. No, the previous one, please. Previous one. Yes. So this is actually what I'm talking about with respect to the general idea about that how we can get the different types with this antibody-mediated protection after convalescence, antibody-mediated protection after vaccination, and effectiveness of therapeutic antibodies. In these three cases, if we look at these variants, United Kingdom, South Africa, Brazil, I'm not talking about the last one, the 1.617, we could see there is always a reduction in this antibody-mediated protection after convalescence or protection after vaccination or of therapeutic antibodies also. And in case of therapeutic antibodies, it was found that in case of the South African Brazil strains, these are ineffective. Next question. Next slide. So the double mutant, which has got the 2 mutations, which has been spotted in fast-spreading variants in California, can reportedly increase the binding power of the virus spike proteins with human cells, making it more transmissible, and can also potentially increase the viral replication. So the E484Q is reportedly similar to the E484K mutation found in the UK. So E484K is a major immune escape variant, and vaccines are therefore less effective. This variant has now long been detected in several countries. Okay. Now, detection be right now, very recently, we could see the triple mutant, the B1.618. So a deletion was also there, a deletion plus 2 changes in the spike protein, H146 and Y145. And the same E484K is there, and there is a D614G. And it was found this is mostly circulating in West Bengal. So the double mutant was done havoc in Maharashtra. You had seen earlier what happens to Maharashtra. Now the Bengal, recently it's been identified the triple mutant. So proportion of SARS-CoV-2 lineages in temporal genome databases from West Bengal has been shown like this. And you can see that proportion, though B1.617 is the main, but B1.618 is also increasing, which is dangerous. Next. Next one. So if the virus evolves so rapidly, is it ever going to be possible to develop modified vaccine fast enough to combat with emerging variants? So this is actually the one. With a global sequencing effort on war footing, it's definitely possible. Next. So distribution of the changes observed in the evolving SARS-CoV-2 region has been shown over here. You can see this is actually the diversity, and virus mutations and variants are being monitored from the start of the COVID-19 pandemic through the global initiative on sharing avian influenza data sequencing database. So through that, it is possible to actually do this, and it is possible to find out that how actually it is going on. So the distribution of the changes which are observed in the evolving SARS-CoV-2 genome is becoming very, very important. Next. And the emergence of the SARS-CoV-2 variants in India, it has been shown by this. You know that the major is a yellow red one. You can see that it's covering in the huge numbers. But the yellow variety and this particular one, which is the green variety or the blue variety or the navy blue variety, these are actually what is actually really the rate estimate is around 8.35 E 4 subs per site per year. And that there actually by it is becoming so important. So the red variety to this one. Next slide. So a similar effort can create a COVID-19 vaccination success story also. The vaccine against the influenza virus are developed twice every year in order to keep the evolution of the virus. So an advisory group of experts convened by the WHO Global Influenza Program analyzed biannually influenza virus surveillance data generated by the Global Influenza Surveillance and Response System, GISRS, and issues formal recommendation for the composition of influenza vaccines for either northern or southern hemisphere influenza seasons. Similarly, the high-yield candidate vaccine viruses are developed by collaboration of laboratories involved in developing the reassortants and WHO collaborating centers. Once developed, these candidate reassorts are sent to WHO, and you can identify the genetic properties before being released. And thereby, we talk about this essential regulatory laboratories in collaboration with vaccine manufacturers. Next slide. So we are looking at evolution-proof vaccines. So a vaccine that comes up with the rapid evolution of the SARS-CoV-2 needs to have the following weapons in its armory. One, it should be able to inhibit viral replication. When the virus stops replicating, it cannot be transmitted and it cannot evolve. The second is the vaccine should induce a multi-pronged immune response that targets multiple epitopes on the virus simultaneously. Studies with antibody cocktails have already proved the strength of this approach. And the third one, these vaccines at the time of deployment should be equipped to target all the currently detected variants. So every day, the vaccine manufacturers and everybody is getting more and more. It's a challenge for them, actually. Next one. So thereby, I end my lecture and thank you, everybody. I'm ready to take the questions.
Thank you, sir, for such an insightful session and new updates from clinical, epidemiological, virologic, as well as pathologic perspective of the virus. We got to understand how the coronavirus protein acts as a multi-molecular machine and mediates the coronavirus entry into the host cell, the different immunological responses, and finally, a very important strategy for designing the vaccination to cope with the highly evolving coronavirus strain. Now, before going to the question answer session, now I would like to request all the participants who are watching us online, it's either through Zoom, YouTube, or Twitter, to fill up the feedback form for today's session. To issue the e-certificate for this webinar, we need your details. Okay, so we expected, as we expected, the chat box is flooded with the question. However, due to time constraints, we will be able to discuss only a selected one. So, Professor Chattopadhyay, can we start?
Yes, please.
Okay. So the first question that appears from, why are we prone to COVID even after the first and second dose of vaccination?
Yes. So, you know that normally after the first dose of vaccination, the immune response, the circulating antibodies are not high enough. And only after the booster dose, we could see that the antibody goes up in the neutralizing antibodies. Okay. So unless we have that in general, because you can see that it's an immunosuppression is there in this particular viral infection. So after the first infection, when the antibodies are still not of that particular stature, you are still going to get your second infection. But what happens, this second infection actually acts as a booster, and the second infection helps to accelerate this antibody production. So for them, you can have the infection even after the first vaccination, but its severity will be much less. In the case of the second dose taken, right after the second dose has been taken, the system is actually geared up for having the production of the vaccine at an accelerated path, as well as the cell-mediated one, the memory cell, they are getting ready with it. So once it goes to the memory cell, you will not get that as a neutralizing antibody in the circulation. But before that, there is a possibility the immune system, when it is very busy with production of antibodies, in general, that is a little bit of what we say that it's similar to the immune suppression also, in other words. So even after second dose, right within 14 days, there is a possibility, unless that 14 days is going to give rise to the total antibody production as well as your cell-mediated one. These 14 days are very crucial, and that time you have to take precautions also. So after that, you are pretty much safe.
Okay, so sir, the next question is, how does the DRDO's new anti-COVID oral drug 2-deoxy-D-glucose or 2-DG help in combating against COVID-19 disease, And will an oral drug be more effective than vaccines?
See, you know that for the cancer patient treatment, this type of drugs are already been there. Because why? The proliferation of the cancer cells are much higher than the regular cells. So the rate, so this use of the different energy is much higher for the cancer cells than the normal cells. So now if we add the 2-deoxyglucose, then what will happen? It is actually not given rise to their energy production. And if the energy production goes down, the cancer cells which proliferate faster, they have a problem. Over here, the similar strategy we are talking about, that the viral replication needs energy. So if you actually, the virus when replicating at a very fast rate in the infected bodies, than the regular replication of the cellular replication process, it is much faster. So they will get the dearth of energy and the viral replication will be slowed down. And in that way, it may work out. But here, I just wanted to mention, yes, it is there, but I am a little bit skeptic about, because here, the negative effect of this 2DG is also very important, because it will affect the normal cells also. The 2DG is not only going to infected cells, but it is also going to the normal cells. So in general, it will actually, the metabolic pathway is going to be affected with it. So maybe to get a bigger disorder, we can afford to have it. But I don't think ultimately it can, in my opinion, it may be the correct choice of the drug.
Right, sir. So, sir, another interesting question that is coming up very frequently, like many companies are taking different strategies to make the vaccines. Like we have RNA vaccines now, DNA vaccine is coming. We have wholesale virus inactivated vaccines. So some confusion is there among the common people. Like, will it be effective to take more than one vaccines or change the brand? And what is that the decisions we should take?
See, the first and second vaccine should be from the same one. Otherwise, the boosting effect is not going to be there. So it is better not to take the different one. It is always good to take the same one.
Right.
But ultimately, we don't know, because after 6 months, 8 months time period, we really don't know what's going to happen or whether a new vaccine is going to be. You know that, as I mentioned, that's why I mentioned about the influenza one. The influenza, this particular committee, which gives every 6 months new vaccines. Why? The reason is that to get rid of the new variants also, so that the people are getting accustomed with it. Okay. So, and in that case, after this second booster dose is over, then waiting for 6 months and get another vaccine to actually counteract with these other strains may come, maybe important actually. We have to wait for that. Immediately, we should not jump on it.
Okay, sir. Would you like to accept one or two questions?
Yeah, no problem.
Okay, so
it is your time.
Yeah, okay, sir. So another question that is very frequently coming is that if PCR detection is somehow less effective than detection of COVID than CT scan or chest, or we fully depend on PCR report only, because sometimes results are negative in PCR through later show positive. I mean, though later it is showing positive. So how this PCR detection is effective?
So try to understand, as a microbiology, virology, other students, we have to understand the PCR test is a very effective process. But where from you are taking the sample? You are taking the sample from the nasal or the swab. So if the virus already traveled that portion and gone into the lungs, then what will happen? The test will be negative. So this new variance was found to reside in our nasal or the throat for a very short period of time. It goes fast into the lungs. So for them to do the RT-PCR, say waiting for 6-7 days and doing the RT-PCR will not give you the positive analysis, isn't it? We are not doing the PCR with this bronchial swab, isn't it? It's not right. For them, the CT scan and other things are more important. So PCR test for these new variants are sometimes may give you negative results, but for them, the CT scan is going to be complementary. Okay. But the PCR test is very important.
Yes.
Or
Yes.
already
Right,
we
sir.
have seen the first phase of infections.
Right, sir. Right, sir. So we almost come to the last question. Sir, can you please shed some light on the black fungus infection that is coming, that is now being associated with COVID-19? Is it something to do with COVID-19?
See, this black fungus infection is actually the system when it is there. You know that what happens when the cytokine storm, we generally treat the patients with the steroids. And when the people are going to be actually look after with the steroids and others, it actually brings down certain immune system. Okay. And
Okay. Yes.
this actually helps this fungus to develop into the system. So
Yes, sir.
normally the protection, that's why always we are telling that whatever medicines you are taking, that your system is how reacting to the steroids and what type of steroids you have to take and what's the dose of steroids you have to take, how long you have to take. All these things are very important to take the medical advice so that the medical practitioners are going to give you proper, oh, dexamethasone is really acting very nicely, so let us take that steroid. That should not be the case, okay?
Right, sir. Right.
So
sir.
rampant use of these things are actually giving rise to this fungus infection.
Yeah, right, sir. So we got a glimpse of update on different aspects of this pandemic. So we have almost come to the end of the session. Now I would like to request Professor Nilangshu Das, the HOD Molecular Biology and Bursar of our college, to deliver the vote of thanks.
Thank you, Devdeep. Am I audible?
Yes.
Yes, sir. Yes.
Okay, it fills my heart with joy unspeakable to cast vote of thanks on behalf of IQAC and the Department of Microbiology, Surendranath College, for a frontier scientist, powerful orator, excellent teacher, and an efficient administrator, Professor Dhrubajyoti Chattopadhyay, Honorable Vice Chancellor of Sister Nivedita University, Kolkata, our very own DJC sir. Sir, you have been supporting us since the inception of this department. I express my heartfelt gratitude to you for giving us time from your busy schedule. Sir, from your lecture today, we got enlightened about the innate and adaptive immune response and the systemic cytokine storm leading to respiratory organ failure. We got to see the very recently published proteome and interactome data, which would help us to design drugs in future. Your presentation about the vaccine took us to the new world of the requirement of rapid genome sequencing. Sir, I express my very sincere gratitude to you again. I would also like to thank our participants from all parts of the globe who were virtually present today with us and made this session a success. I hope your experience was satisfying. My gratitude to our Honorable Principal, Dr. Indranil Kaur, and the governing body of Surendranath College for being a constant source of encouragement. Thanks are also due to IQAC Surendranath College, all the faculty members of the Department of Microbiology, and especially the very efficient technical team for their crisp handling of the event. Finally, I express my love and respect to all my colleagues of Surendranath College, teaching and non-teaching, and our very beloved students present and past for being with us all throughout this webinar. I'd like to conclude by requesting everybody to stay at home as much as practicable, observe extreme precautions, and strictly obey all the guidelines framed by central and the state government. Together, we will fight out COVID-19. Thank you. Thank you very much.
So this brings the end of our today's session. We will be bringing you a series of new sessions from the desk of scientists and physicians who are directly involved in the research of deadly virus. So stay tuned and do join us every Wednesday in our webinar series. With this, me, your host Devdeep, signing out. See you all in the next session on 19th May, right at 3 p.m.
Thank you, Devdip. Thank you, Nilangshu. Thank you, everybody, and definitely the principal of the college. Thank you.
Thank you, sir. Thank you very much.
Thank you, sir. Thank you very much, sir.
Thank you. Thank you.